Good morning. Hope everyone is safe and doing well. My name is Bill and I'm a biological scientist from University of Florida, Gulf Coast Research and Education Center. My topic today is using biostimulants to increase tomato yield in Florida. Florida is the biggest producer of fresh tomatoes in the United States. There are many approaches discovered so far to improving fruit yield and quality, and one of which is using agricultural biostimulant. Biostimulants are substances or microorganisms that could be applied to the plant and stimulate the natural processes to improve their nutrient uptake, stress tolerance, and in some cases, the quality of the crop, such as nutritional content, shelf life, etc. If we compare fertilizers as the plant food, which supplies the essential nutrients, including macronutrient and micronutrient, then biostimulant is the supplement, which increases the availability and uptake of the nutrients. Some common types of biostimulants include humic acid, fulvic acid, amino acid, crab shell powder, seaweed extract, algae, and mycorrhiza. Today I'm going to share the results of 2 trials that we conducted for companies in which biostimulants showed some positive results. In the first trial, we applied humic acid and seaweed extract. Humic acid is the final breakdown components of the natural decay of organic materials. It binds to plant root system and increases soil cation change capacity, phosphorus availability, and the nitrate uptake by stimulating nitrate transporters. The seaweed extract has hormones that promote plant growth and has anti-stress effects by providing antioxidants and regulating stress-related genes. Usually, seaweed extracts are applied in 2 forms, either a liquid extract or a powder. The trial was conducted in the spring season of 2019 at Gulf Coast Research and Education Center in Baum, which is in Central Florida 9B on the plant hardness zone map. We used a popular fresh market cultivar, HM1823. Treatments were performed by injecting the chemical through a dosatron injector, and control received the same amount of water. Experiments followed a randomized complete block design with 4 replications. 5-week-old seedlings were transplanted in February, and there were 2 harvests in May. We focused on plant canopy area, fruit yield, and shoot biomass among different treatments. The picture on the right is an example of how we perform canopy area data collection. First, we took overhead photos of the plot, then processed image using ImageJ software. For fruit yield, we collected data from 8 representative plants of a plot and followed the same commercial standard to grade the harvest, in which the unmarketable fruit is anything lower than 100 grams in weight or has any defect. Marketable fruit could be subcategorized as medium, large, and extra large, depending on the fruit size. Here are some photos showing plant growth at different timings. Running the statistical analysis, we didn't find any treatment effects on the plant canopy area or shoot biomass. but we did find a significant difference in marketable yield. Using humic acid or seaweed extract product, we were able to increase the yield by 10%, although the percentage of marketable fruit yield is not affected. Moving on to the second trial, in which we applied a mycorrhizal product. Mycorrhiza is a symbiotic association between plant and fungus. Plant provides organic molecules to the fungus, and the fungus makes water and minor nutrient more available to the plant. This drawing on the right side of the screen is showing the difference with and without mycorrhiza. Plant with mycorrhiza has better access to the nutrients and thus has a stronger root system. As a result, it could better support the canopy and benefit the plant as a whole. The field trial was conducted in the fall season of 2018 at the same location using the same tomato cultivar as the first trial. The mycorrhizal product was applied using two methods. Number one, spray application before transplanting, like the picture on the right, showing spraying the product in a greenhouse with a CO2 backpack sprayer. The second method is the field drenching that happens immediately after transplanting. The experiment followed a randomized complete block design with 4 replications. 4-week-old seedlings were transplanted in August 2018, and fruit were harvested twice in November. Apart from canopy area, shoot biomass, fruit yield, we're also interested in which method of application could establish mycorrhiza more efficiently. The mycorrhizal colonization was measured by the company using light microscopy. Here are the results. Compared to control, greenhouse spray and field drench had higher rate of mycorrhiza colonization, which is not surprising, but only drenching application is significant, suggesting it is the best practice to apply the product. In terms of canopy area, although at the beginning of the season, drenching application has bigger and denser foliage, this effect didn't last until the end. As you can see, the results are not significant near the harvest. As for shoot biomass and marketable yield, no significant difference could be observed, although drenching application increased the yield by around 10%. Because the trials were conducted for the companies, I am not able to review the details of the product formula, but we can see that certain types of humic acid, seaweed extract, and the mycorrhizal product could have some beneficial impact on the growth of the tomato and the fruit yield. Field drench is the best way to establish mycorrhizal colonization. I want to give my thanks to the lab personnel for their hard work and also the cooperative companies, and see you in the Q&A session. Thank you.